So, Bismillahirrahmanirrahim. In this video, we will now discuss the prominent process of inflammation, that is recruitment of the leukocyte to the site of inflammation. So, up till now, if you were following me, you know what is inflammation. It is the response of a vascularized or a normal tissue to a pathogen, a microbe, or to the necrotizing or necrotic tissue. Firstly, what happens that the microbe is recognized. We have done the recognition of the microbe in the previous videos that you can watch in the playlist of this Robbins Pathology. After the microbe or the necrotic cell has been recognized, we recruit the leukocytes from the circulation to the site of injury to eliminate that microbe from the site of injury, or to eliminate the dead tissue or debris from the site of its presence. So in this step, in this video, mainly we will focus on the recruitment of leukocytes. So leukocyte recruitment to the site of inflammation. Why is it done? To eliminate the offending agent that is present at the tissue. Okay, so as you know, if you've been following through my videos, that in acute inflammation we have the the cells that are recruited, they are neutrophils, whereas in chronic inflammation the cells that are recruited, they're either lymphocytes or they're either monocytes, and monocytes are then activated into the macrophages. So these neutrophils in the acute inflammation, where do they come from? They come from bone marrow. So as you know, they are formed from the bone marrow. So what do these neutrophils do? They do cytoskeleton arrangement as well as enzyme assembly, and in this way they help to eliminate the offending agent from the tissue where the pathogen is present. So the neutrophils, why are they involved in the acute reaction? Because they are rapid and they show transient response, immediate response. That is why they are involved in acute inflammation. Why are lymphocytes and macrophages involved in chronic inflammation? Because they are slower to come to the site. They have prolonged responses. They are slower, but they produce more potent and more prolonged responses. That is why they are involved in chronic inflammation. And the effects through which they do this is by gene transcription, by affecting protein synthesis in the offending agent, whereas in acute inflammation it is done by cytoskeleton arrangement or by enzyme assembly. So this is the major differences that you can write between them. Now what do the macrophages do? They engulf and they digest the agent or the microbe, as well as they release growth factors, and these growth factors do the repair. So they not only engulf and digest the pathogen, but they also help in repairing of the tissue or returning it back to normal. But only drawback is that these neutrophils and these macrophages, sometimes they do tissue damage, they do collateral damage to the normal tissues. Whenever they attach to the cell that we will study further, that is known as margination, in that margination process they can damage the endothelial wall of the cell. That is why they have a tissue damaging drawback. Now the journey from the vessel lumen to damaged tissue, it is mediated and it is controlled by adhesion molecules and cytokines. What do I mean by this? I will explain everything in detail, but first look at this diagram. So this is a vessel. Vessel is blood vessel. So blood vessel has all the cells that are flowing through and a little bit fluid around it. What will happen now, that leukocytes will be recruited in such a way that the red blood cells, they will move in the center, whereas the neutrophils or white blood cells, they will move to the sides. When they will move to the side, they will attach to this vessel wall. After attachment, now they will migrate through these intraendothelial pores. That migration is known as transcytosis, and after that migration they will follow the pathway that is given to them by chemoattractants or chemokines, and thus they will do the chemotaxis to site of injury. So what do they do? Firstly, they were moving in a streamline, but now because they have been recruited, they arrange themselves in such a way that red blood cell in center, white blood cells to the sides. White blood cells, they move through these pores, the intraendothelial pores, that is transcytosis, and then they follow the pathway given by chemokines to the site of injury. Now we will discuss how the leukocytes adhere to the endothelium. So what is endothelium? Endothelium is the vessel wall. Vessel wall, it is made up of endothelial cells, so we call this whole thing endothelium. So first thing first, that I told you that the red blood cells will come to the center, whereas white blood cells will arrange them to the side. That process is known as migration. What happens? Blood is normally flowing in a laminar flow in the capillary and in the postcapillary venules. But what happens that when we want, when there are cytokines released, when we want the recruitment of the leukocyte, what happens in capillaries, especially in postcapillary venules, that these red cells, they arrange themselves in the center because they are lighter, whereas white blood cells, they are heavier and they arrange themselves to the periphery. So this is how margination takes place. After that, we know some amount of fluid starts to leak from the vessel wall, so stasis develops. What is stasis? In the previous video we have discussed as well that fluid leaks out, the cells are more in number, so the cells become slower, and this is how stasis develops. Now these are hemodynamic changes, and wall shear stress decreases in such situations. So the main crux of this is that red blood cells in the center, white blood cells to the side, and then these white blood cells will attach to the vessel wall. Now, how will they attach to the vessel wall? For the attachment, the vessel wall has to project these adhesion molecules. There has to be expression of the adhesion molecules. First of all, these white blood cells will have loose attachment. That loose attachment is that the white blood cell is over here, it will bind to this adhesion molecule, then it will roll over to the other one, then it will roll over to the other one. This is known as rolling. So first step was margination, red blood cells in center, white blood cells to the side. Then because of the presence of certain cytokines and other mediators, the cell wall or the vessel wall starts to express these adhesion molecules. Then after margination we have rolling process, and after rolling process, when cell or when the neutrophil or the white blood cell finds a proper molecule that is made for it, it adheres to that molecule, and that is known as firm attachment. So the whole process that is followed is margination, red blood cell to the center, white blood cells to the side, rolling. How does rolling take place? Because of the expression of adhesion molecule, the neutrophils roll over and then they adhere to specific sites where their adherence is possible. So margination, rolling, and adherence. This is how leukocytes adhere to the endothelium. Now in the next thing, we will discuss what is rolling process. The rolling process that I was telling you, that the cell membrane, the endothelium, it expresses these adhesion molecules, and the neutrophils roll over from one molecule to another molecule till they find something for their firm attachment. So now the adhesion, the rolling thing, it is in such a way that leukocyte, it binds, then detaches, and then binds again, and this is how it rolls over. Now there are selectin molecules which are expressed on either the leukocyte or on either the endothelial cell wall, and in such a way they help the leukocyte to roll over. So there are three types of them. There are E-selectin, L-selectin, and P-selectin, and they have this particular name as well, CD6, that is the CD62, that is the prefix, and then similar, E-selectin has E to it, P-selectin has P to it, and L-selectin has L to it. So E-selectin is also known as CD62E, L-selectin CD62L, P-selectin CD62P. Now they can either be expressed on endothelial cells, this one, or they can either be expressed on the leukocyte. So E-selectin, as the name suggests, it is coming from endothelium. So it is present on the endothelial cells. Similarly, L-selectin, it is present on leukocytes, whereas P-selectin, it is present on platelets and endothelium. Now the thing that you need to recognize over here is that selectins are receptors, and receptor needs ligand. For example, if E-selectin is present on endothelial cell, for example, it has a receptor present in endothelial cell, so where will be its ligand present? Its ligand will be on that leukocyte. You see? So E-selectin has a receptor on the endothelium and ligand on the leukocyte. Similarly, L-selectin, it is expressed on leukocyte. So if the leukocyte has L-selectin, so where will be its ligand? Ligand will be on the endothelium. Same goes for P-selectin. It is the receptor will be on the platelet, or endothelium. So if we make it here, the receptor will be on the endothelium, but where will be its ligand? The ligand will be on the leukocyte. So this is how you need to remember the whole thing: E, L and P selectin. Now the ligands for these selectins, they are sialic acid containing oligosaccharides with a glycoprotein backbone. So for example, this is a glycoprotein backbone and it is a sialic acid containing substance. So it has a glycoprotein backbone, but it is a sialic acid containing substance, that is the ligand. Now we can talk about these p-selectins. They are saying that the p-selectin or that receptor, it is not expressed until it is needed. And when does the need arise? The need arises whenever there is tissue injury or they are upregulated by cytokines. Okay, so where are they stored in such a situation? They are stored in Weber palate bodies. And whenever there is histamine or whenever there is thrombin released, they express themselves, these purple p-selectins express themselves on either the endothelial walls or on the platelets. Similarly, E-selectin and L-selectin, they are also stored in somewhere, but they are released or they come to the surface whenever there is cytokines, interleukin 1 or tumor necrosis factor alpha released. So for P-selectin, we have histamine or thrombin. For E and L-selectin, they require a stimulus like interleukin 1, TNF, and cytokines, the cytokines that are coming from the resident cells, that are mast cells, dendritic cells, and phagocytes. We have discussed this in the previous video with the whole diagram. You can consult that diagram from Robbins and you will remember each and everything. So now we have this whole thing where I have shown that what happens to the endothelium in such a situation. Because E-selectin is a receptor that is coming on the endothelial cell, so it will be expressed on the endothelium. But L-selectin, since it is present on the leukocyte, so endothelium will express its ligand. So this is how the whole thing occurs. That E-selectin is expressed on the endothelium, but ligand for P-selectin is expressed. So now if we have the final comparison, so the endothelium will be expressing this one, the brown one, that is E-selectin, the receptor. Then it will also be expressing the P-selectin, which is also the receptor, but it will be expressing a ligand, which is the L-selectin. Ligand for L-selectin. But when we come to leukocyte, leukocyte will express one receptor only, that is the L-selectin, and it will express two ligands, ligand for P-selectin and ligand for E-selectin. And all of these will be expressed on the tips of microvilli. I hope you understood. And these adhesion through these receptor and ligand interaction will allow the leukocyte to roll over the endothelium from one place to another place. So this is how it takes place. Now, why is this rolling important? This rolling is important. This attachment, detachment, which is known as rolling, it is important because it slows down the leukocytes and gives additional time to respond to the adhesion molecules, so that better quality leukocytes are selected to be thrown out of the vessel wall to the site of injury. So this is what happens. Now we have to go for firm adhesion. After rolling, we have firm adhesion. Now firm adhesion, it is stimulated or it is regulated by integrins. What are integrins? Integrins are transmembrane 2-chain glycoproteins. Selectins were receptors, whereas integrins are transmembrane 2-chain glycoproteins. Now what happens with firm adhesion? Leukocytes, they stop rolling. They signal the cytoskeletal changes. They arrest the leukocyte and they allow firm attachment of leukocyte to endothelium. When they are firmly attached, then what happens? That the intraendothelial space is now open and then they allow the leukocyte to move through. So rolling is important, but after rolling, firm attachment is also important. Rolling was done by selectins. Now firm attachment will be done by integrins, which are transmembrane 2-chain glycoproteins. Transmembrane means they run through the membrane, and 2-chain means they are having 2 chains. So I have made the whole thing transmembrane 2-chain glycoproteins. Now they mediate the adhesion of leukocyte to the endothelium. They're mainly because they are transmembrane, so they will be present in leukocyte plasma membrane. The location is very important. The leukocyte plasma membrane will be the location of integrins. Now they are also present, they are also expressed, but when are they expressed? They are expressed or they are activated by chemokines, which are chemoattractant cytokines. What do they do? For example, this was your integrin, or this was your leukocyte. It was having this integrin, but this integrin was it in its non-activated form. Whenever a cytokine comes, that cytokine or that chemokine, which is a chemoattractant cytokine, it activates the integrin. The integrin is expressed on the surface of the leukocyte, now fully expressed, and that integrin now recognizes the ligand for the integrin. The ligand is present on the endothelium, and hence it firmly attaches to the endothelium. So how it needs to be activated? It needs to be activated by chemokines. The chemokines bind to endothelial cell proteoglycans. This chemokine, this orange thing is the chemokine, and that is actually present on the endothelial cell proteoglycan. Whenever that rolling thing, that rolling leukocyte, it falls over this chemokine which is present on the cell membrane proteoglycan, what will happen? It will fully express the integrin. The fully expressed integrin will now bind to the ligand, and hence we will have firm adhesion. Now there are different types of ligands. For integrin we have ICAM, we have VCAM. So because I told you that ligand will be present on the endothelial one, so I have written the ones with orange, that is ICAM and VCAM. Now this ICAM, it's translated at intracellular adhesion molecule 1. That intracellular adhesion molecule 1 attaches to two things that are present on the leukocyte, that are LFA1 and MAC1. So we have two things, ICAM, VCAM. The ICAM attaches to two things further, which are L and M, that is LFA1 and MAC1. Both the LFA1 and MAC1, they are expressed on the leukocyte. The LFA1, it is CD11A, CD18, whereas the MAC1, it is CD11B, CD18. The whole name is same, but one is A, one is B. The LFA is A, the MAC is B. Now this LFA is translated as leukocyte function associated antigen 1, whereas this MAC1 is macrophage 1 antigen, or macrophage antigen 1. So the main thing was that the ligand is ICAM or VCAM, but the receptor for the ligand is, for ICAM, the receptor are L and M. L is LFA1 and M is MAC1. This one is the one with A and this one is the one with B. For VCAM, which is a vascular cell adhesion molecule, we only have one receptor, and that one is VLA4, that is very late antigen 4. So you can recognize the name. ICAM, VCAM, they are the ligands. For I, we have L and M, LFA1 and MAC1. For VCAM, we have only one, that is very late antigen number 4. So this is how firm attachment is regulated for the leukocyte. After the firm attachment, what will happen? The leukocyte will now move through the intra-endothelial pores, but we will discuss the movement in the next video, inshallah. I hope you understood each and everything about rolling, about firm adhesion that was explained in this video. I did my best level thing to explain the whole scenario in this video. If you like my style of teaching and my content, please like, share and subscribe, and we will meet in the next video, inshallah.